Okay, let's start anyway. Today our topic is general virology, and we start our last part in microbiology course. From this time, we will talk only about the viruses and the viral infections. Today, I would like to introduce the virology to you because virology it's one of the most difficult a difficult part in our course. So the viruses, as you know, it's filterable agents, and it was first described as a filterable agents because it's critically small, and the viruses can pass through the bacterial filter. It's exactly about how Dmitry Iosifovich Ivanovsky, Russian botanist, discovered the first virus in the world. It was not human virus, it was tobacco mosaic virus, and he discovered in 1892. He actually worked with tobacco plants and he filtrated a smash of tobacco leaves through the bacterial filter and inoculate the filtrate to new plant, and the new plant became sick the same disease. It get the same disease, and he found something much smaller than bacteria can infect. Of course, right now we know much more about the viruses than he knew, and right now we know few general things about the viruses. First of all, they are obligate intracellular parasites of all kind of cells. So if you will found anywhere alive, you can be sure the viruses are also present in this environment, sample, whatever. Uh, Over the world, we have much more usually viruses than organisms. One organism can be infected by various types of viruses. So, about the viruses, we can say few general things. First, very small filtratable agents, obligate intracellular parasites. Viruses cannot make energy or proteins independently on the host cell, so they need host cell. Viral genome may be RNA or DNA, but never both. Instead, our cell, instead bacterial cell. Viruses have a naked capsid or an enveloped morphology, which I will tell you carefully a bit later. Viral components are assembled and do not replicate by division. So viruses never replicate by division. We told about several bacteria which are also obligate intracellular parasite, but they are always replicate by binary fusion. Here not. Viruses never do that. They use absolutely another mechanism of replication. Generally, viruses are not living, but it can be looks like living organism when they are inside the cell. Viruses must be infectious to endure in nature. If virus can't infect the cell, it will die anyway. Viruses must be able to use host cell processes to produce their components. So, what the major components of the virus? Usually genetic material, some enzymes, and glycoproteins. Also, viruses must encode any required process not provided by the cell. And virus component must be self-assembled. So this assembly usually it's the last stage of viral replication. Sometimes it happens inside the cell, sometimes it happens outside of the cell, but it always independent on the cell. So there are just several steps when the virus can do independent on the cell, and we will talk about all of them. So to talk about virology, of course, we have to say a few words about classification. This is a classification what I found in one of very common book for medical students, book in microbiology. But I don't want to give you such big classification. I would like to introduce to you very small classification of viruses, but very useful for us. Look here, please. There are major components of the virus on the top. You can observe. So the viruses can be classified depend on these components, presence or absence of some of them. And later here you can observe 5 columns. dsDNA means double strain DNA, ssDNA single strain DNA, plus single strain RNA, minus ssRNA, and one more time plus single strain RNA but another type. So also viruses can be classified depend on its nucleic acids, depend on its genetic material. Viruses range from the structurally simple and small parvoviruses to very big and very complex pox viruses or herpes viruses, what you can observe here in the left column. Their names may describe viral characteristic, the diseases they are associated with, or even tissue or geographic location when they are first identified. For example, name retrovirus, which you can observe in the right column. Retrovirus, the member of Retroviridae family causes HIV. Retro because reverse. Reverse to the virus detected synthesis of DNA from an RNA template, whereas the pox viruses are named for disease cowpox caused by one of its members, pox virus here. So, as you see, virus is primarily classified by its genetic material, but also, as I said, by the structure. Let's say a few words about. So, the structure. What's important in structure? Size and morphology of it, nucleic acids, as I said, biochemical characteristic, also important for classification. Structure and mode of replication can be also described and be based for putting the viruses in one family or into another one. Disease, for example, encephalitis, hepatitis, it's a major example. You know, hepatitis viruses can cause hepatitis A, hepatitis B, hepatitis C. All of them are totally different. All of them can be found in different families. Some of them totally independent viruses. But anyway, they are in one group because they can infect one organ, its liver, and of course the cells of the liver hepatocytes. Means of transmission, arboviruses spread by insects, that's why we call all of them arboviruses. And host cell, for example, animal for human viruses or mouse, birds, some plant viruses, and bacteria viruses we know as bacteriophages. We told about this group last semester. Sometimes tissue or organ tropism also used as a base for that. For example, adenoviruses and enteroviruses are grouped because infect tissue or organs. The first group, what we have to say, it's DNA viruses, and for all DNA viruses, we also can divide to enveloped viruses and naked capsid viruses. As I said, I will say about it later, about enveloped and about naked viruses. Right now, just observe the major groups of them, major families. Pox, herpes, hepatinoviruses, all of them are enveloped. Polyoma, parvoviruses, papillomaviruses, adenovirus, DNA naked capsid virus. So, actually, as you see, there are only 7 families which are associated with human diseases. So, DNA viruses was. Next big group, RNA viruses. First of all, RNA viruses should be divided on the type of RNA, positive or negative RNA, or double positive and negative, or retroviruses, as I said, specific group. So we can find at least 13 families of RNA viruses which are medically important. In this group, you can find some viruses what you already know. As I said, retroviruses, and of course, right now everybody know coronavirus. Coronavirus is an enveloped positive sense RNA virus. Next, what we have to discuss about, it's about viral structure. What does it mean on the picture? How to understand what are the spikes, what is the curved things and the dots inside. About that, I will say later. So, please look on the right column. It's a relative characteristic of the size and the shape of a major group of viruses. Usually, viruses very small, but sometimes it can be pretty big. And the smallest viruses, which are important for in medicine, it's parvoviruses because it's only 80 nanometers. And the biggest one, it's a pox virus, which pretty big, 300 nanometers. 300 nanometers, for example, it's a chlamydia bacterium elementary body, what you already know. Look here, chlamydia and pox viruses look similar by the size, but it's not mean it's very similar by its structure or nucleic acids, but of course then bigger virus, then more components it can include, so large genome, some enzymes inside, of course, then bigger container, then more carriage we can put inside. Usually big viruses also very complex. They have much more not only inner structure but actually external structures. All viruses invisible under the simple light microscopy. We can observe it only under electron microscope. And you can see here, it's an transmitted electron micrograph of T4 bacteriophages which are attached bacterial cell, and some of them already injected its own genome inside bacterial cell. You can observe it here. It's exactly genome strain. To understand what does it mean naked and enveloped virus, we have to understand what exactly component virus could have. First of all, as I said already, virus should have DNA or RNA. Okay, let's choose some of them. Choose DNA, for example. Add structural proteins Sometimes we have to add enzymes, and it means we have nucleocapsid which equal naked capsid virus. It's very simple. So take a DNA, put it inside um, protein structure, and you have naked capsid virus. To make enveloped virus, we have to surround all this structure of naked nucleocapsid virus by glycoproteins and membrane, and we will have enveloped virus. So such kind of viruses looks much more complicated, but please understand, glycoprotein and membrane which around it, it's not a shield. It's absolutely not a protection. Usually, such kind of virus is much more sensitive for environmental factors than naked viruses. So it's not a shield. It's just one more layer. Okay, look here. Non-enveloped viruses has, as I said, only genome and protein capsid. Sometimes inside also some enzymes are. Enveloped virus, instead naked, has polyphospholipid bilayer. This kind of phospholipid bilayer has a very complicated structure, and it's totally depend on the viral genus, so its own characteristic. Different kind of viruses has different uh, components in the envelope. Two major definition what you should understand: virion and virus. Virion is the infectious particle. What you can observe on the picture, it's exactly virion. So when we're talking about the bacteriophage T4, it's also virion. So all these complex structures are virion. Virion may be intracellular or extracellular, doesn't matter. It's an infectious particle. But instead, virus, it's any stage of infection. Yes, you can say virus when you observing non-enveloped or enveloped virus. You also can say virus when you're talking about current infection, means virus not looks like this complicated structure. It's just a genome. Anyway, it's a virus. So virus, it's like a stage of infection. Few words about naked capsid. So all non-enveloped virus, as I said, very simple by its own structure. But what's not simple here? Not simple, it's because difficult to kill such viruses. They are very stable for temperature, for acids, proteases, enzymes, detergents, and drying. Such kind of viruses can live on the surfaces for years sometimes. Moreover, they are released from the cell by lysis, means death, cell death should happens to produce new viruses. So all that these viruses replicate itself inside the cell and then just destroy the cell and go out. As I said, they very resistant to environmental factors. That's why they can be spread by some vomits, hand-to-hand shaking, and dust, small droplets, fecal oral route also here. Uh, Dry out and retain infectivity. It's important for them because if they have to survive in dry environment, they have to be active anyway. They can survive the adverse conditions of the gut, so stomach acid barrier can't stop them, most of them, of course. They can be resistant to detergents and poor sewage treatment. and antibody may be sufficient for immunoprotection. It was about naked capsid virus. Next few slides will be about enveloped viruses. Look here. All of them complex. So not only nucleic acids inside, not only proteins, but also glycoproteins on the surface, lipids, and the membrane. Moreover, usually enveloped viruses has enzymes inside. So the complex structure not means more resistant than non-enveloped viruses. Usually they are environmentally labeled. So acids, detergent, drying, and heat can kill them very effectively. Usually they release out of the cell by budding, sometimes by cell lysis. So enveloped viruses can go out of the cell and cause cell death or not, because their envelope include membrane, but they can't make the membrane by itself. They have to take a piece of membrane from infected cell. So like coat, make a coat from infected cell. Excuse me, I don't understand who are talking. Okay, I see. Представитель деканата, не могли бы вы звук выключить? Выключите, пожалуйста, свой микрофон. Okay. Let's continue. So, all these viruses must stay wet. If they are not wet, they reduce the infectious possibility. So, that's why they always must be inside tissue or liquids. That's why it can survive in the gastrointestinal tract because of acids in the stomach can kill them. That's why they spread only in large droplets, secretion, during organ transplantation and blood transfusion. Does not need to kill the cell to spread. Means virus can stay in human body for very long time, slow reproduction, and avoid cell killing usually cause latent infection when the person has the infection but don't know about it or have no symptoms of it. Also, they may need antibody and cell-mediated immune response for protection and control, elicits hypersensitivity and inflammation to cause immunopathogenesis. So the virion envelope is composed, as I said, many kind of structures. It has membrane structure similar to cellular membranes. Sometimes it's exactly a part of cellular membrane. Cellular proteins are rarely found in the viral envelope, even though the envelope is obtained from the cellular membranes, as I said. So it's depend on the type of the virus. Some viruses make it by itself. Some viruses make this similar to the membrane structure, and some viruses can take it exactly from the cell. Most viruses, envelope viruses, are found around or pleomorphic. Two exceptions are the pox virus, which has a complex internal, a brick-like external structure. Uh, if you saw the picture before, the brick, it's exactly how does pox viruses looks like. And the rhabdoviruses, which is a bullet shape. I will go back to show you it once again. Here is a member of pox viruses, brick-like structure, and rabies viruses. But actually, it's a section of rabies viruses. It's a half of rabies virus, and all the rabies virus has bullet shape. Anyway, we will talk about all these viruses in our near future during a couple of next lectures. Uh, Another key, what we have to say, it's a symmetry of the capsid. Symmetry of capsid also important characteristic of the virus. Each virus has its own specific structure, and here on the micrograph picture, you can observe different symmetry of the and different capsomers. Capsomers, it's a pieces of the envelope of the virus of the protein. The capsomers, it's the smallest structural proteins which bind to each other and which make a protein coat. Next important function. On the picture here, you can observe the major steps in the viral replication. I will talk about viral replication several times, and I will show you different pictures of it. But anyway, all any kind of pictures has similarities, because viruses, doesn't matter what kind of virus, has very similar replication cycle. Only small differences will be described later. So the major steps in viral replication are the same for all viruses, as I said. The cell is used as a factory, and the virus control this factory to produce its own viral protein and replicate its own genome. Process not provided by the cell must be encoded in the genome of virus. So if virus will lost a control, virus will die. Virus can lose the control because lost some piece of gene. Such kind of infection we call abortive infection, when the virus infect the cell and then can't do nothing. So will die anyway. The manner in which each virus um, accomplish this step and overcome the cells biochemical limitations is different for different structures of the genome and of the virion. So we have several major steps. First, recognition of the target cell. Second, attachment. Third, penetration. Next, uncoating. Fifth one, macromolecular synthesis. And actually, it's one of the most complicated because it's differences between different kind of viruses. Genome will replicate it by itself from the matrix or from the template or not. Actually, structural protein synthesis also could start rapidly from positive RNA or after several steps. Post-translation modification of proteins also could happen in these big steps. So, macromolecular synthesis. Next, when all components of the viruses are synthesized, assembly of virus will happen. After that, depend on the situation, virus can just release or budding, depend on the virus. If it's enveloped, it will bud. And is release. So let's talk about how could it happen. On the picture you can observe major steps, but please understand, I will show you. So first step, recognition, and second, attachment happens for all kind of viruses, but attachment could be different. For naked viruses, attachment just like happens, the attachment and penetration looks like phagocytosis. But if the virus has envelope, attachment happens by different scenario. Attachment to the envelope and the cell happens, and then fusion happens. So all components of the viruses like fuse inside the cell, but never make uncoating in this step. So uncoating step happens for naked viruses, primary. Then depend on the type of the viruses, transcription, protein synthesis, and replication is happen, and assembly happen for all all naked viruses, and then lysis and release happens. If the virus has enveloped, assembly happens after budding. So the envelopment happens nearby the membrane, then budding happens because of the membrane will surround the virus, viral components, and budding. After that, full assembly will happen. On the same picture, you can observe a different kind of chemicals or immune components which can stop each step in viral replication. For example, recognition and attachment can be stopped by antibody receptor antagonist and also by several drugs which has a target on the surface of the virion. Then, Then, many kind of drugs can prevent inner steps, fusion uncoating, transcription, synthesis of protein, replication also. Moreover, protein synthesis can be prevented by our own interferon. And protease inhibitors also can stop assembly when the virus still inside or it's outside of the cell, doesn't matter. Also can be prevented. Here you can observe different nucleic acids, different not cycles actually, different ways how does virus will replicate itself depend on its own genome. First picture A, positive sense single strain RNA virus. So plus SS RNA viruses. All of them has similar kind of way. Look here. After attachment and penetration and uncoating, single-strain RNA can be used as a template to produce viral proteins. Look here. Single-strain RNA directly used as a template for synthesis, protein synthesis, and it's used as a mRNA. So another strain or the same strain after division can be used as transcription by the viral RNA polymerase and complementary negative sense RNA to act as a template to make a copy of positive single strain RNA. After that, all components will assemble. Next scenario, negative sense single strain RNA virus, so minus ssRNA. Because it is minus, why we call it minus? Because it can't directly be used for translation. Can't totally. Usually, it need transcription by RNA-dependent RNA transcriptase enzyme to make a complementary positive RNA, which will act as a template and as a mRNA. So, translation of viral proteins happens from this copy of positive RNA, and also further transcription will make a copy of negative sRNA, which we'll use for assembly together with viral proteins. And the last scenario, it's a double-strain RNA virus. Double-strain RNA virus has positive sense and negative sense. Positive sense act as mRNA. Negative sense act as a template and transcription by viral RNA polymerase to make complementary RNA strands. So both of them will be assembled together, winding, and assembly together with viral proteins. So as you see, it's not difficult. Just positive can be used directly for transplantation. Negative should make complementary positive RNA. Double-strained has positive and negative both together. Different kind of genome very influence on the viral life cycle. Double-strain DNA usually need specific, each stance of DNA serves as template for its complement. And of course, it should produce RNA. Only exception here, it's a hepatitis B, which synthesize RNA to act as the template for new DNA. Double-strain, it was double-strain DNA. For single-strain DNA, mRNA synthesized by RNA polymerase in nucleus of cell. And complementary strands of DNA is synthesized to act as a template for genome replication. For positive RNA, I told, and for negative, I told, and for double strain, I told, but I didn't say nothing about Retroviridae family, which is specific and single strain RNA can be used as a template for new, because mRNA should be synthesized by DNA from DNA, because first of all, RNA viruses will be transferred to DNA, then it will be back transferred to RNA. So such kind of uncommon way happens because of reverse transcriptase enzyme, which can transfer RNA to DNA, but not like usual process from DNA to RNA transcriptase used. And of course, such kind of virus very specific. We will talk about HIV virus specifically. We will have specific class for it, separated class, and I will tell you everything about viral replication in this case. Right now, just please understand it's uncommon because DNA converted from the RNA, but not back like normal process. So, as I said, non-enveloped viruses will just destroy the cell, make the cell lysis and go out. But enveloped virus very clever. They use sometimes cytoplasmic membrane of the host to go out. If they use cytoplasmic membrane of the host, they could be sometimes invisible for the immune system because membrane usually include our own antigens, which shouldn't be recognized as a foreigner antigens. And of course, sometimes it's like a mask for them. But anyway, viral capsid can attach new cell without specific proteins on its own surface, and the spikes usually can be recognized as a foreigner substance. Let's take a short rest for 5 minutes, and after that we will continue to talk. We have couple of subtopics, and first we will start to talk about viral diseases, of course briefly. Rest for 5 minutes. Let's continue. Tell me, can you hear me? Can you hear me
Yes,
well?
sir.
Yes, we can.
Yes, sir.
Yes, sir.
Very good.
Yes, good.
Yes, sir.
So, good. About viral diseases. As you see on the picture, all kind of cells, organs, and tissues of our cell can be infected by the virus. So there is no any kind of cell which can't be infected by the virus. Anyway, the viruses, as I said, very, very educated, and the evolution of the viruses goes on the strict way, infect everything. So, and they do that. In our classes, we will discuss about many viruses which infect a lot of different organs, and our classes will be grouped on the specific diseases which viruses cause. So, the major sites which virus can infect, it's the respiratory tract. The most common viral infections over the world, it's a respiratory infection because easy to spread, easy to infect because nearby, and we all of us we are breathing and we can't keep breath, so you have to breathe, and with air you also uh, consume viruses. Gastrointestinal tract also important because a lot of infections, viral infections, can be transmitted by the same way, but it's not mean it cause infection of intestinal tract. It's can go deeper and can transfer into other organs. A very nice example here, it's poliovirus, which use gastrointestinal tract as a primary site of infection, but do not cause any kind of symptoms here, but then infect nervous system. Epithelial cells, mucosal, endothelial, of skin, mouth, and genital organs, lymphoid tissues also can be infected. Of course, liver and other organs, inner organs can be infected, and of course, central nervous system. Moreover, our immune system cells can be infected by the virus, and the very good example here, HIV virus, which infect T helper cells, which should protect us from viral infections, but no, virus infect them. Of course, our immune system trying to solve the problem, but it's not always possible. Um, Viruses, viral diseases can be grouped by site of infection, can be grouped by the way how it's transmitted to human body, but sometimes it's not very difficult to understand what kind of site of infection is a major site, if virus can infect different kind of cells, or the cells, similar cells can be found everywhere. But anyway, we trying to identify what's exactly a major one. A major one, not only inside of infection, also major transmission way. You know, the viruses can be transmitted by different ways. The last one, coronavirus. You know, the primary way for transmission it's airborne, but it also can be transmitted through the handshaking, yeah, and then leaking. We call it contact way. So, of course, the major way here it's airborne way, but we would say we can identify and classify this virus because it's a respiratory one, or we can classify this virus because it's very dangerous and put it in group of highly dangerous infections. So doesn't matter. You will understand everything what we will talk about. I will try to explain you anyway. So several diseases either produce symptoms or have epidemiologic or other characteristics that resemble those of viral infections, or maybe the sequel of viral infections. inflammatory responses, or to a persistent viral infection. They include multiple sclerosis, Kawasaki disease, arthritis, diabetes, and chronic fatigue syndrome. So, such kind of problems we also will discuss, but shortly. Our primary key point will be exactly the virus. What is it? And also we will talk about the immune response. As you know, the strong immune response happens because of cell immune response and of course by antibody immune response. If you remember, we have specific group of cells which can recognize viral infected cells and destroy them, T killer cells. They can protect us very well from the viral infections. Also, in our classes, we always will talk about laboratory diagnosis. And laboratory diagnosis of viruses can be very various. First of all, to talk about lab diagnosis, we have to talk about cultivation. Viruses can't be cultivation on the artificial media, so they need live cells. How to make the comfortable conditions for the viral cultivation? First of all, we can make specific media, which include live organisms. For example, bacteria, plants, or animal cells. Then we can take embryonated eggs and inject the viruses inside. And the last one, we can use cell cultures. So, cell cultures, it's usually a very novel method, but let's talk about it one by one. First, culturing viruses in bacteria. We know about it because we did it. Sorry, I told about it. Usually we are doing it in the class, and we know this method as bacteriophage typing. We use that. So we use bacteriophage and bacteria as food for them. Actually, of course, not food, as a factory. Next, culturing viruses in plants and animals. So live laboratory animals can be used to cultivate viruses inside them. Embryonated egg. Actually, the virus will grow in an embryonated egg, and this can be a very convenient and inexpensive form of host for many animal viruses. A hole is drilled in the shell. You can observe how it's possible, usually by needle, in the shell of embryonated egg, and a viral suspension or suspected virus containing tissue is injected into the egg fluids. There are several membranes in an egg, and the virus is injected near the one of the most appropriate for its growth. Viral growth is signaled by the death of the embryo, by embryo cell damage usually, or by the formation of tubercle pox or lesions of the egg membranes. This method was one of the most widely used methods for isolating and growing viruses. And it's still used to grow viruses for some vaccines, for example, influenza vaccine, primary influenza viruses primarily cultivated in the eggs. So for this reason, if you are allergic person, it's better to ask about vaccine, because egg proteins may be present in the viral vaccine preparations. So If you allergic for the egg component, it's better to ask about the vaccine before do it. Last way for cultivation is the culturing viruses in the tissue culture or cell culture. Cell isolated from an organism and grow on the surface of a medium or in broth. Antibiotic usually injected inside to limit the growth of bacteria. Cell cultures are two types. First, diploid cell culture from embryonic cells and continuous cell culture from tumor cell. So continuous cell culture very nice because they never die as all tumor cells. So apoptosis is not present in this cell, not programmed in this cell, and of course such kind of cells are very nice to grow viruses. To understand, does virus grow inside the cell or not, we can use cytopathic effect of viruses. So cytopathic effect, it's specific changes which happens after viral cultivation in the cell. Look on the picture. Uninfected human cells, cervical cells, each has one nucleus. It's picture A. But after infection with herpes human virus 2, the red cervical has many nucleus. They are, I'm sorry, they are usually fill it with the virus and make it bigger. Another variant of the cytopathic effect, it's inclusion's body in brain tissue from a person who died of rabies. Inclusion's body, or we call it Negri body, very visible on the tissue. Portion of syncytium, or the giant cell, you can observe on the picture B. Such kind of giant cell formed in the cell infected with measles virus. The cytoplasmic mass is properly the Golgi complex or fused cell, but anyway, the cell is totally big with many nucleus inside, syncytium, because many cells bind each other and combined in one. Okay, we understand how to cultivate viruses. Let's talk about identification. Viral identification, it's a process, of course you understand it's an important process because we need it for the diagnosis, primary for diagnosis of infection. We need to know what kind of virus caused the infection because usually it's totally influence on the treatment strategy or epidemiological situation. So first of all, not first of all by importance, but just first in our list, identification of virus in cell culture. Second possible way, microscopic identification directly in the specimen. Third possible way, serological procedures to detect a rise in antibody titer or the presence of IgM antibody, just presence. Fourth way, detection of viral antigens in blood or body fluids. And the last possible way, but not last by importance, it's detection of viral nucleic acids in blood or the patient cells. I would like to tell you few words, just few words about each way for identification. First, viral growth in cell culture frequently produce a a specific cytopathic effect, what I already told about, that can provide a presumptive identification, just presumptive. If the virus does not produce a cytopathic effect, its presence can be detected by the several other techniques. For example, hemadsorption. Hemadsorption is an attachment of erythrocytes to the surface of a virus-infected cell. This technique is limited to viruses with the hemagglutinin protein on their envelope, such as mumps, parainfluenza, and influenza viruses. Second technique, interference with the formation of a cytopathic effect by a second virus. For example, rubella virus, which does not cause cytopathic effect, can be detected by interference with the formation of a cytopathic effect by certain enteroviruses, such as echovirus or Coxsackie virus. So, means cultivation two viruses with one cell. So it's increased the possibility of the cytopathic effect visibility. And the last technique, it's decrease in acid production by infected dying cell. This can be detected visually by a color changes in the phenol red, which is a pH indicator, in the culture medium. The indicator remains red alkaline in the presence of virus-infected cell, but turns yellow in the presence of metabolizing normal cell as a result of the acid procedure. This technique can be used to detect certain environments, enteroviruses, and in environmental also. Next way, it's a definitive identification. It's possible by several tests. Usually, we use it when the growth in the cell culture is made by using no antibody. So, first of all, complement fixation test. You know about it. We told about it last semester a lot. Next one, hemagglutination inhibition, neutralization, fluorescent antibody, radioimmunoassay, enzyme-linked immunosorbent assay, which you know as ELISA test, and immune electron microscopy. So all these techniques we can use to identify viruses in cell culture. Next, viruses can be detected and identified by a direct microscopic examination. So we can use clinical specimens such as biopsy material or skin lesions and just put it under the microscope. So there three different procedures can be used. First of all, light microscopy can reveal characteristic inclusions body or multinucleated giant cells. So it's a kind of cytopathic effect we can observe exactly in the specimen from a person. Second procedure, UV microscopy is used for fluorescent antibody staining of the virus in infected cell. So anyway, we will observe specific effect under the microscope by naked eyes. Sorry, by naked eyes, of course, by the microscope. And last procedure, it's electron microscopy, which only can help to observe viral technologies, and we will detect DNA or RNA of virus inside specimen. If we did it, means it's a viral infection. And that's why it's becoming a gold standard of the viral diagnosis. Usually, by some techniques, we not just can detect presence or absence, we can count how many viral particles are present in 1 milliliter of blood, for example. It's important characteristic for the prognosing and the monitoring of the diseases. And answering for the question, does a treatment produce successful results or not? So that's it about the viruses for today. And I would like to ask you, have you any questions about our lecture or any other related questions to me? If you have any questions, please stay here. Let's talk. If you have no any questions, goodbye. See you 2 weeks later. Next lecture will be about acute respiratory viral infections. We will talk also about coronavirus.
Okay, sir.
Okay, sir. Bye, sir.
Goodbye.